Hi folks, I'm here to highlight some of the scientific method and experimental design work that we've been sharing in classes. So first off, this is a series of 3 scientific experiments that are historically important in biology on a topic known as spontaneous generation. So long time ago, hundreds of years we're talking about, um, people believed that life could come from non-living things. We know that in order to generate more organisms, you have to do reproduction. But prior to our understanding now, we thought that life could come from non-living things. Case in point, the belief at the time that maggots came from rotting meat. Now, Reddy's experiment on spontaneous generation challenged this. This is a great highlight of the scientific method because the first process that you do is you identify the question. All right. So, do maggots come from rotting meat? Whenever you write the question, it has to be in question form. Then, because Reddy, the scientist in this particular experiment, was trying to solve this or answer this question, he had a hypothesis. He believed, and this is evidenced in the way he designs his experiment, he believed that maggots came from flies that land on the meat. Well, how is he going to test this? He tested this experiment by setting up a series of glass jars with rotting meat in them. 2 of the jars were left open. 2 jars were sealed, and 2 jars had some gauze over the top. Gauze is just a fabric with holes in it so that flies would be able to smell the rotting meat there. And so he put these different jars in the same location with the same types of meat and for the same amount of time, same size jars, everything needs to be the same except for the one variable. We identify this as the independent variable. So in this particular experiment, where you have all these little flies flying around the rotting meat, the one variable between all these jars is the presence of top or gauze covering. All right. It was either left open, it was completely sealed, or it had gauze on it. Now, you might be thinking, well, I thought you were only allowed to have one independent variable. That is true. There is only one variable in this case, and it's the covering on the jar. Either there's no covering, it's sealed covered, or it's got the gauze cover. The dependent variable is the variable that we measure in our experiment. So Reddy's experiment, he was looking for the presence of maggots forming on meat. In the first set of jars, where they're open to the air, flies were able to land on it and they were eating the meat, and while they're doing that, they're laying their eggs there. Now, they didn't know the eggs were being laid there because the eggs are microscopic, so you wouldn't see them there. But they do develop into maggots, which is why people believe that the maggots came from the meat. A control group is one group of your experiment setups that is used for comparison. It is not manipulated. The one setup in this case that is not manipulated or changed by the scientist is this first group with no top. All the other groups are known as experimental groups, and the controls are those factors that are kept the same. Temperature, it could be type of meat, it could be size or type of jar. Everything about these has to be the same, or the results will be inconclusive. Now I'm going to bring you to a second experiment. And this one right here, because one of the premises of the scientific method is that when you publish your results, other scientists need to be able to evaluate your work. So this second experiment, because scientists said, well, wait a minute, what about, just because you didn't have maggots grow in that middle jar doesn't mean that they didn't spontaneously generate, because where did they come from before that? Scientists are going to try and punch holes in your experimental results. So a new scientist came up and said, well, wait a minute, so microorganisms still exist because, frankly, even if I have something in Tupperware in my fridge, mold can still grow on it. So even though Reddy had sealed the middle jars, mold grew on them. And so Spallanzani did a separate experiment to try and help disprove the idea of spontaneous generation. All right, and in his case, he developed one with a flask with gravy in it that was boiled. Now he did this with two different flasks, both with the same amount and type of gravy, heated them both up to boiling, and then one he left open, and the other one he melted the container to seal the top shut. So he melted the glass and sealed it. All right, so now I'm going to ask you to do the following. Pause the video, look at this picture, and see if you can identify the question, the hypothesis, independent and dependent variables, the control group, and the controls that are kept the same. Pause it now and see if you can do that, because I'm going to go over it together with you in just a moment. Now, assuming you paused it and you checked it out, what is the question that he's asking? In this case, you might say do microorganisms come from gravy? And your hypothesis might be, the microorganisms come from the air and grow there. Well, if that's the case, your hypothesis has to be testable. So you've got to boil the gravy. Why would they boil the gravy? Because that would kill any microorganisms. The one independent variable in this case is the sealed jar, or excuse me, flask, or open flask. It's either sealed or it's open. All right, that's the one variable. What are they looking for as a response? What is the dependent variable? Presence or growth of microorganisms. So in this first setup, which is the control group, flask left open is the control group for comparison. This is what would normally happen. Even though it was boiled to kill any microorganisms, they grew in it afterwards. All right. And the results of the experimental setup showed that the gravy is free of microorganisms. Nothing could grow in there. The controls include, hopefully you had, things like type and amount of gravy, size of flask, temperature that it was heated to, where it was stored. All these things have to be controlled, because if you change anything, someone's going to say, well, how do you know it's because the flask was open? It might have been because you kept one in a dark room and one in a light room. All right, so the results seem to be conclusive. No microorganisms could grow there. But the problem is that scientists and society at the time believed in something called phlogiston. Phlogiston was this miraculous material found in the air that provided life for everything. And scientists said, you did not give the gravy in the sealed container the ability to get to phlogiston. So no life could grow there, but life would normally be able to. It was, I can't make this up, it was called dephlogisticated air. So they couldn't conclusively say that the experiment was a success. They had to have some other experiment to develop and support it. And that's what brings us to Louis Pasteur. And this is the ultimate experiment that he designed to answer the question about spontaneous generation. Again, what's the question? Can I should ask you to pause it and see if you can fill these in. So take a moment, pause it, and see if you can fill in this experimental setup. And so here we have, now that we're back, the question that he's trying to answer is, do microorganisms come from broth? And you might even be interested to say, can microorganisms grow in broth that has been sterilized, because he's going to heat it so it's boiling again. His hypothesis is microorganisms come from the air and land in the broth. He developed a really unique gooseneck tube that goes on the top of his flask. And he had only one variable again, but he had just one setup, which was unique. In this case, he put the gooseneck flask or gooseneck tube on the top of his flask and left it there and then broke it off a year later. So the one variable is the curved glass tube on, whoops, this should be glass. The curved glass tube on the flask. The dependent variable is growth of microorganisms again. The control group is when the glass tube is intact and then the experimental group, actually, I just did that backwards. The control group is when the glass tube is removed, and then the experimental group in this case is when he's got this glass tube in place. And what he found out is that microorganisms would fall into this tube from the air, but they don't have the ability to get up and over the top into the broth. So even though it's open to the air, it stays microorganism free because they can't get to the nutrients down there. And it's not until he breaks off the top that they're able to. So the controls in this case, because he only has one flask, everything is the same. All characteristics are the same. All right. Now, in each one of these cases, the scientists had to publish their results, and scientists at the time either accepted or rejected their results as an explanation for the belief of the time. But each one of these was designed as an experiment with a hypothesis. They had to collect observations, either in the form of quantitative or qualitative results. They had to determine or evaluate their data to come up with a conclusion, and then you would have to publish your results. All of these things are part of the scientific process that you need to be familiar and comfortable with, right? So I hope that this helps by using 3 legitimate scientific experiments. Even though they sound like pretty ridiculous, these were real and very important experiments, which is why when you buy milk at the store, you see it as pasteurized on the container. And the reason why it's called pasteurization is because of Louis Pasteur, who designed this process as an explanation for debunking the idea of spontaneous generation. I hope that helped, and if you have any questions, see your teacher.